Hey, what's up, everybody? Welcome to, well, a little bit of work at home. We got protein synthesis, and what we're going to do is we're going to make a video, probably about 10-15 minute video, something like that. Going to make a series of them, probably maybe 4 or 5 per chapter. And from there, you will have some questions that you can address or work on with the Google Classroom, and there'll be a project and we'll figure something out for a test. So here we go. A lot of this first video is going to go quick because we did a lot of this already, so I just want to kind of refresh people's memory. And as we get towards the end of the stuff we didn't do, I'll slow things down a little bit. So protein synthesis is basically taking the genes or the genetic code from the DNA and through a series of making mRNA and then going out to the ribosome, ultimately making a polypeptide chain which will fold and form a protein. So for DNA replication, which we talked about, we know that DNA is double-stranded, and there's going to be a bunch of enzymes that we talked about, and DNA is going to simply take one double-stranded molecule and make an exact copy of itself into 2 double-stranded molecules. That also happens in the nucleus, and as we remember with DNA, A will make a bond with T, and C will make a bond with G. Protein synthesis is a little different. There, you're going to have a series of nucleotides in a row on the DNA, and a specific area there may be called a gene. And what will happen is you're going to take a different enzyme. We're going to take a RNA polymerase this time, and we're going to do a process known as transcription. We're going to make a molecule of mRNA. That molecule of mRNA will then be able to leave the nucleus. So proteins, in essence, what they are long chains of amino acids called polypeptides, and they will not function until they fold. Examples of polypeptides would be enzymes, hormones, receptors, and they can also be structural as well. So those are what proteins are. And proteins pretty much control everything. They're structural, like we said, and they can act as signaling molecules in the form of hormones. Again, they cannot do anything. They are, in essence, useless until they fold. So the instructions to make a polypeptide chain is in the DNA, and those specialized regions are called genes. And the DNA is what is the instructions. So pretty much the only enzyme we're going to need here is an RNA polymerase II. And when we remember DNA replication, we had like 6 enzymes, and they all had a very specific role. Here, RNA polymerase does a lot of things kind of all at once. And its job is to basically start with a DNA strand and code for an mRNA strand. And that's its job. And this is going to happen in the nucleus, and it's going to be a process known as transcription. Transcription, as we know, or as I just said, is taking the code from DNA and making a piece of mRNA from it. In DNA, we know that A bonded with T and G bonded with C. But in transcription, because we're making mRNA, and we learned last time that RNA does not have any T's, it has U's instead. So the A is going to bond to a U, and the G still goes to the C. But the AU is a big deal here. So if you take a look at this picture, this is kind of all in here. But this kind of bubble-shaped teardrop thing, that's your RNA polymerase II. And it does multiple jobs all at once. It's going to take the DNA, it's going to separate it for a second, and then it's going to call in the complementary base. So, for example, if there's a T on the DNA, it's going to call in an A. If there's an A, normally it would look for a T, but in this case, because it's RNA, it's looking for a U. And what you are creating is this red molecule here, which is this, as you can see in the diagram there. That is a piece of RNA, and specifically it is mRNA for messenger RNA. Now, the DNA has 2 strands when you open it up. It has a template strand, and it has what is known as a coding strand, or the non-template strand is otherwise known as the coding strand. Now, what's going to happen there is, once again, the template strand is coding for its complementary bases of mRNA. Now this here is the whole picture, so you can kind of see it all together. You have DNA, which is here, double-stranded. That's where your A's, your T's, your G's, and your C's are. Then that little area right there is where you're making this piece of mRNA right there. And you'll notice it's single-stranded, and it has A, U, G, and C. It will then leave the nucleus. We'll talk more about that in a little bit. But ultimately, your goal is to form this molecule right there, which is a series of amino acids known as a polypeptide chain. All of this building takes place at this grade-in molecule here. This grade-in, not molecule, organelle style thing is known as a ribosome. And again, we talked about that a lot last week when you were in class. So similarities, we know that DNA, we already did this, has A, T, G, and C, where in transcription of making mRNA, you're going to have A, U, G, and C. DNA is a double-stranded molecule, where mRNA or RNA is single-stranded. They both happen in the nucleus, so that's good. So your DNA is in the nucleus, and your mRNA is in the nucleus, but what's kind of cool is this new molecule can actually leave the nucleus, okay? And here you have the deoxyribose sugar, and in the RNA, the transcription, you're bringing in the ribose sugar that comes with the RNA. Okay, so mRNA, once again, as we kind of talked about, is a single strand of nucleotides, and just like all the nucleotides we talked about, it has a phosphate group. It has a pentose sugar, but in this case, it is a ribose sugar, where here you're going to have an OH group, and you're going to have your base, which is right there. I don't know what happened there. Get rid of that. Do that again. You're going to have your base, and your bases are A, U, G's, and C's. mRNA can leave the nucleus, as we said, and begin to form this polypeptide chain. So as we zoom in a little bit further, once again, there's your mRNA. Each of these things right there is a nucleotide. So that could be, let's say, an A, that could be a U, that could be a G, that could be a C, whatever. Once it leaves, it's looking for this grade area there, known as a ribosome. And you're going to begin to build, once again, this polypeptide chain. It is made up of individual pieces, and each of these little dots is one amino acid. And we know if we put together a bunch of amino acids, we're going to get a polypeptide chain. We'll talk about how that goes a little bit later. So let's practice this transcription. So the very first thing you have to remember is, as soon as you see a T, then you know that is DNA, so that's an easy one. And if this is the template strand, that means that you are making the mRNA from that. And from here, if that's a T, that's going to be an A. Normally that A would be a T, but it's going to be a U and a G. And then we're going to go A, C, G, 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 C, C, G, and U, A. And if you've done that, you have done transcription. You made yourself a piece of mRNA that has the ability to leave the nucleus. And once again, you know the top strand is DNA because of the letter T. Now, I'm going to use this word a lot. It's called a codon. Now, a codon is a 3-base sequence of nucleotides from the DNA to the mRNA. So let's say AUG. That could be one codon. It's one codon because it is a total of 3 bases, with 3 nucleotides. Now, each codon is going to, in essence, code for one amino acid. Now, the amino acids, when I say code for them, it means it's going to go get it, because the amino acids are already in the cell. It doesn't make them, it's going to go and get them. So you're going to use something known as the universal genetic code, and you're going to do that when you're making mRNA. So let's take a look. For example, Now I know that this is a codon on the mRNA because I see the letter U. So if I want to know what amino acid that codon is going to go and get, I use my universal genetic code, which is the diagram on the right. So I look for a U, a C, and an A. And I love this chart here. And you know that UCA is going to start with the U right there. Then your second base is the C, and then the A falls in. It's right there. So once you find that codon, you know you can code for the amino acid SER or serine. Get rid of that so we don't get too confusing. Let's try CUC. So CUC is right here, which is going to go get the amino acid leucine. AUG is going to be right. AUG is going to be right there. That's going to go and get methionine or MET. And UGA, UGA, that's going to be an interesting one. That's actually not going to go get any amino acid. That is what is known as a stop codon. So a stop codon is going to become important when we get up to that. Okay, so every polypeptide has to start somewhere and it will eventually end. It starts with what is known as the start codon and the start codon. And here's where you got to be careful. The start codon is AUG. That is going to code for the, in essence, start amino acid of methionine. Okay, so be careful on the question. The codon is the basis. The amino acid is what you will go and get eventually using your chart. And the three stop codons are going to be UAA, UAG, and UGA. So those are your three stop codons. And again, you don't have to memorize any of that because it's going to be on your universal genetic code, which you'll be supplied with. Now let's see what we got. We know once again that this is DNA, and we know it's DNA because of the T's. We have then made 5, or we coded for 5 codons, and they're mRNA, and you know that once again because of the U's. So now if I look on my chart, I know that AUG is methionine, MET. AAC is going to be ASN, which is again an amino acid. And let's see, 3 G's is going to be glycine, GLY. 2 C's and a G are going to be proline. And then UAA is a stop codon. So in this case, my polypeptide chain consists of 4 amino acids. Well, like everything else, here's what it would look like in real life. You need this, the AUG, that's your start codon right there, and your UAG is going to be your stop codon. So this, in essence, will be the polypeptide chain. So you know there's going to be 1 codon, 2 codons, 3 codons, 4 codons, 5 codons. So by the time you're done, you should end up with 5 amino acids. Oh, 6 actually, because this is going to be an amino acid methionine. This one doesn't count as amino acid because it's a stop codon. So there's your ribosome. And your ribosome is going to move along, and as it moves along, that is going to create your polypeptide chain. So let's see what we got. AUG, I already know is met. I mean, kind of memorize that one. That's an easy one. And again, GGC is going to be glycine, GLY. UUA is going to be leucine, LEU. And AAG is going to be lysine, LYS. CAG is going to be GLN. And UGC is going to be cysteine. And finally, that is a stop codon. So if you can practice this, you have just made a polypeptide chain. Well, we kind of talked about this already. Where do amino acids come from? And they come from the food that you eat. That's where you get them from. Or you can recycle old proteins. Pretty cool, right? Again, your cell does not waste anything. Well, how are they brought to the ribosome? Well, they're going to be brought to the ribosome by a molecule known as tRNA. So once again, the amino acids in the cytoplasm came from the food you ate, and the way they get there is your stomach and digestive system digested them. So now they're nice and small. They float in your bloodstream. And then once they get to a cell, they're simply going to diffuse into the cell. Because at this point, the amino acids are pretty small, and they're going to diffuse in. And how do they get to the ribosome? They're going to be brought there by a molecule known as tRNA. So what happens is the mRNA goes out, it goes to the ribosome. It's going to attach. Once it attaches, this cool molecule known as tRNA, it's kind of like an Uber, and what it does is it brings specific amino acids to the mRNA, which is at the ribosome. As the mRNA moves through the ribosome with the help, as it moves through the ribosome, you need some ATP to help move it. And from there, you will keep on calling for a new amino acid, a new amino acid, a new amino acid, and this again will happen until you reach what is known as a stop codon. You will have a polypeptide chain at that point that will eventually fold, and once it folds, it will function. And translation is what we just did. Translation is the act of bringing the amino acids that are floating in the cytoplasm to the ribosome, and it's done with the help of a tRNA molecule, kind of like an Uber. Now, a tRNA molecule it's kind of funky, and once again, you know that it's RNA because of the U's, and it's I can't even really describe what it looks like, kind of like those things, those jacks, that game jacks where you spin that little metal thing and bounce that ball, maybe, if you guys even know what that is. But what's going to happen is the amino acid is going to attach here. So whatever amino acid you have, that's going to be attached. Now, how do you know what amino acid you need? Well, this little thing called an anticodon, that's going to bind up to the codon. So let's say, for example, this is your mRNA. And let's say you have, I don't know, 3 U's. U. Now, we know that 3 U's is going to get phenylalanine, PHE. But it's got to make a temporary junction with this tRNA molecule. And the tRNA, the anticodon, is going to be 3 A's. Now, once again, do you have to get hung up in that? No. But you do have to know that that will make a temporary junction with the codon, allowing the tRNA to stick for just a second to drop off the amino acid. Once it drops off the amino acid, the tRNA can leave and go find another amino acid. So here it is. There's that beaker-looking thing right there. That's your tRNA. That little black dot's the amino acid. And you can see that the tRNA is coming over and joining with the mRNA. That right there is your anticodon. And that anticodon is going to make a temporary match right there for just a second. And what will happen is this will get, actually it kind of goes this way. That will get added to the amino acid that was brought over. Then this tRNA can leave, and it just happens until you reach a stop codon. Again, it's pretty amazing. So circle a codon. So codon would be here, and you know that would because that's on the mRNA. And the anticodon, let's get rid of this since I circled it, but let's square it. The anticodon is right there, which is on the tRNA. So let's kind of build this protein and watch how it goes, and then we'll be done with video 1. So you have the ribosome, which you know is right there. You have your what happened over here? You have your mRNA, and you know that because of the U's, and it's single-stranded. And what's going to happen is, you know that you're looking for your first codon, which is AUG. Well, boom, there it is. You just brought over your methionine. This UAC, that is the anticodon. And what'll happen is, so those are your amino acids, your tRNA molecule, which you now know about. There's your anticodon, which you now know about. So what's going to happen is the next codon of GGC is going to be looking for the amino acid GLY. Well, there it is. Now notice the tRNA did not get there yet, but it's going to. And once it does, it's going to make a temporary junction right there. Now at this point, the methionine and the GLY and the glycine, they're not attached yet, but they will be. And you're going to form something called a peptide bond. And again, I'll show you how to draw that in the next video. But a peptide bond is going to form, and there it is. So at this point, that first tRNA can leave. And when it leaves, the met and the gly will be joined. And there it goes. Bye. And what's going to happen is this will continue to move down. So the met and the gly is still together. We got to call in our next amino acid. So UUA is going to get leucine. And there it is. Once again, it didn't get there yet, but it will. Once it does, you have to form a bond. And once you form that bond, right there, and once again, if you can't see it, these are your peptide bonds right there. Sorry if I wasn't clear. And now that tRNA that was there can leave. And this will continue. As this goes on, you will build what you just made is your polypeptide chain. Once again, it is not a protein yet. It will be, but not yet. Until eventually you're going to reach that thing right there. And that we know is a stop codon. And once you hit that stop codon, everything's going to let go. So let's watch what happens. Once you hit that, once again, there they are. Your 3 stop codons are right there. Once you hit any one of those 3 stop codons, everything breaks. Ribosome goes one way, that last tRNA goes one way, and now, believe it or not, can you actually reuse this if you want to? Question mark, and the answer is yes. Can this ribosome be reused? Answer, yes. So that's what's nice about this process. You don't need to keep on making new mRNAs every single time. You can just keep on repeating it over and over and over again. And again, once again, everything's going to re-enter the cytoplasm. Now the protein is complete, and once it's complete and it's been released, you're going to learn, well, you're probably not going to learn in this class, but different amino acids have different charges, or they have different what's known as hydrophobic or hydrophilic qualities, and that will allow them to fold in a very specific pattern that will eventually allow them to fold and function. So that's kind of what we did already. That's the kind of big first video there. The rest of the videos will be shorter, but I just wanted to kind of wrap everything up. So I will be posting this with instructions and questions to follow. All right, see you next time.